All right, today we're going to zoom way in, deep into a world that's totally hidden from us. It's a world of these tiny biological machines that have been shaping life on our planet for, well, billions of years. We are talking about viruses, and we're going to break down exactly how these invisible invaders manage to hijack our very own cells.
So take a look at this. It looks just like a little lunar lander from some old sci-fi movie, right? But what you're actually seeing is a bacteriophage, a virus that specifically hunts bacteria. And here's the wild part. It's not really alive. Think of it more like a precision-engineered machine built for just one single, relentless purpose.
And that purpose really gets to the heart of what we're talking about. What if a machine's only goal was to make more copies of itself? Because honestly, that is exactly what a virus is. It's like a piece of software, a biological program that can't run on its own. It absolutely needs a computer, in this case, a living cell, to execute its code.
And that, my friends, makes it the ultimate biological hacker. So let's dive into how these microscopic intruders break into a cell's operating system and basically turn it into a factory for making more viruses.
So what exactly is a virus, when you get right down to it? Well, the official definition calls it acellular, which is a fancy way of saying it's not even a whole cell. It's just this tiny infectious particle that is completely 100% dependent on a host. Why? Because it has no metabolism, it can't make its own energy, can't build its own proteins. It's the ultimate parasite, basically just carrying the blueprints and the tools to break in.
Now, once a virus finds a target, it doesn't just have one plan of attack. For these bacteriophages, there are two main playbooks they use to take over a cell, and we can think of them as smash or sneak.
And these two strategies could not be more different. First, you have the lytic cycle. This is a pure smash and grab. It's fast, it's brutal, and it's totally destructive. The virus gets in, forces the cell to make copies, and then just blows the whole thing apart. But then you have the lysogenic cycle. This is the sleeper agent. The virus slips its code inside, and that code hides inside the host's own DNA, just waiting, lying dormant for the perfect moment to strike.
Okay, let's look a little closer at that smash and grab. It all starts with attachment. The phage physically locks onto the host cell. Then comes penetration, where it injects its DNA. That's the moment the hijacking begins. In a stage called biosynthesis, the cell is completely taken over and forced to churn out viral parts. During maturation, all those parts get assembled into hundreds of new viruses until finally, lysis, the cell just bursts, unleashing a whole new army of hackers onto the world.
Now for the stealth approach, the lysogenic cycle. Here the phage still infects the cell, but instead of an immediate hostile takeover, its DNA actually integrates itself into the host's own chromosome. This little piece of hidden viral DNA is called a prophage. And now, check this out. Every single time the host cell divides, it copies the viral DNA right along with its own. The virus is literally hiding in plain sight. Then, maybe the cell gets stressed out, and a trigger called induction happens. The prophage activates, cuts itself out, and boom, it kicks off the destructive lytic cycle.
So that whole smash or sneak playbook, that's for viruses attacking bacteria. But what about the ones that come after us? Well, the principles are pretty similar, but the targets, our cells, are a little different. So it's time to meet the rogues gallery of human viruses.
You see, our cells are different from bacteria. We don't have that rigid cell wall. So viruses have to get in either by being swallowed up by the cell, a process called endocytosis, or by just fusing directly with our cell's outer membrane. And you know, viruses are total specialists. The flu virus, for example, is built to attack the cells in your respiratory tract. Poliovirus, on the other hand, goes after the brain and spinal cord. This specificity has a name. It's called tissue tropism.
Let's pull up a case file on a very familiar foe, influenza. So the flu virus attaches to a cell lining your airway, and then the cell itself actually pulls it inside. The virus's genetic material, its RNA, is released. And in a really unusual move for an RNA virus, it travels all the way to the cell's nucleus to get copied. After that, new viruses are assembled, and then they're released by budding off the cell surface, all ready to go infect their neighbors.
But some RNA viruses are even more cunning. They're called retroviruses, and they carry this special tool, an enzyme called reverse transcriptase. This little enzyme allows the virus to do something that's supposed to be impossible. It completely rewrites the central rule of biology. It takes its RNA code and works backwards, turning it into DNA. And that DNA can then be permanently stitched right into our own genome.
And of course, HIV is the most famous example of a retrovirus. It fuses with one of our immune cells, and then reverse transcriptase gets to work, turning that viral RNA into DNA. That new DNA then integrates into the host's chromosomes, becoming what's called a permanent provirus. Now, this might sound a lot like that bacterial prophage we talked about, but there's a huge difference. This provirus almost never leaves. The cell is now, for all intents and purposes, a permanent factory for making more HIV. This ability to hide out brings us to this idea of latent infections. The varicella-zoster virus is a perfect and probably familiar example. First, it causes chickenpox. But even after you get better, the virus doesn't actually go away. No, it retreats into your nerve cells, where it can lie dormant for decades. Then, years later, it can reactivate and cause that really painful rash we all know as shingles. It's the ultimate sleeper agent.
Now, figuring all of this out took decades of painstaking research. But think about it. How do you even begin to study an invisible enemy that can only replicate inside a living cell? Well, this question leads us to a remarkable and very complicated human story. The story of the immortal cells of Henrietta Lacks. You see, to fight viruses, you have to be able to grow them in a lab. And to grow them, you need host cells. But for the early virologists, there was this one massive problem. Human cells were just incredibly hard to keep alive in a lab dish.
This process of growing cells outside an organism is called cell culture. And the big roadblock was something called contact inhibition. It's actually a natural safety mechanism. When most of our healthy cells bump up against each other in a petri dish, they just stop dividing. It's like they know the space is full. And this meant that any cell culture scientist tried to grow would die out pretty quickly, making long-term research pretty much impossible. Then, in 1951, came the breakthrough. Scientists discovered that the cervical cancer cells from a patient named Henrietta Lacks did something no other human cells had ever done in a lab before. They just kept dividing, and dividing, and dividing. They were, for all practical purposes, immortal. They had lost that contact inhibition, which, ironically, made them the perfect host for viral research.
But behind this incredible scientific breakthrough lies a really profound ethical story. These cells, which are now famous as HeLa cells, were taken without Henrietta's knowledge or her consent. Her family had no idea they even existed until the 1970s, a full 2 decades after she had passed away. It wasn't until 2013 that her family finally gained some control over their genetic data, and only in 2023 did they reach a settlement with a company that had profited from her cells. It's a really critical and complicated chapter in the history of medical ethics.
So the story of HeLa cells allowed us to understand viral replication on a cellular level. It was a game changer. But to really, truly understand how any of this works, how a virus knows which cell to infect, how a drug can possibly stop it, we need to zoom in even further. We need to get down to the fundamental principle that governs all of biology, molecular shape.
This brings up a pretty basic question, right? How does a virus actually recognize the right cell? How do all these tiny, tiny parts fit together so perfectly? The answer isn't just in the biology, it's in the chemistry. Specifically, the 3-dimensional shape of molecules.
Okay, this brings us to a concept called enantiomers. Now stick with me here, it sounds complex, but it's really simple. Just think about your hands. They're mirror images of each other, right? But you can't lay one perfectly on top of the other. A left-handed glove will not fit on your right hand. Well, enantiomers are exactly the same idea, but for molecules. Same chemical formula, but they're mirror images with a totally different 3D shape.
So why does this matter so much? Well, the thalidomide tragedy from the 1950s is the most powerful and, frankly, tragic example. This drug was sold as a mix of its 2 enantiomers. One shape, one hand, was a safe and effective sedative for morning sickness. But its mirror image, its other hand, was a teratogen. It was a molecule that caused absolutely devastating birth defects. A tiny mirror image difference in 3D shape had horrific real-world consequences.
And that's what it all comes down to, really. Shape. Viral proteins fit into our cell receptors like a key fitting into a lock. An antiviral drug has to have the perfect shape to block that lock. It's this whole world of intricate 3-dimensional puzzles. And that leaves us with one last thought. If a single mirror image molecule can cause such devastation or provide such a perfect cure, what other secrets are still out there just hidden in the shapes of the invisible world all around us?